So before I started my talk, I'll briefly introduce my background. I got back from Taiwan, and then I decided to do research. So I applied for a PhD program at CMU, biomedical engineering. Most of my research are doing MRI. Started with brain MRI. My PhD is cardiac MRI. But in my PhD training, what I did is try to correlate MRI finding with pathology. And that's why we have communication with a lot of pathology people, and that's why I'm here. So today's talk will be a lot of things will be from the radiology side, but some of the material will be related to pathology because it's a great trend that people want to validate the things you look at, want to validate the finding you see on the imaging. So there's a lot of collaboration between two sides, between pathology and also radiology. So just a brief introduction of the topic. So I will briefly introduce something called the human connectome. So you will see the topic. There's a new term called connectome here. This term may be new to some of you, but I guess most of you heard of genome. Genome is like the genetic sequence. Connectome is like a kind of a coding or diagram of how brain wire each other. So as we show here in the figure to the right, these things people call it connectome. Is that you see a human brain. If you view each brain region as a node and try to see whether regions are connected to each other and plot those diagrams, then people call it connectome. So this term was created 10 years ago by computational biologists. They thinking of a way to map how brain connect to each other. But it's a lot of work because in the past we can only see gross anatomy when you overlap human brain, and what you see is gyro, folding, sulcus, all the structure here. But we have no idea whether those two regions are connected to each other. The trick here is because the connection between regions are epsilon. Epsilons are at the scale of micron. You cannot visually see how epsilons connecting from one region to another. So if there's a way we could plot the epsilon trajectory, which we call the chartography, then we could build up something we call connectome. And a lot of scientists believe that if we can map human connectome, then we have key to know how brain works or not works, how brain function or dysfunction, and find out what's the normality of the brain structure and also the abnormality. So it's really a key to a lot of neuroscience people and also clinical people working on neurology, psychiatry, people interested in psychology. They would like to do that. So this is really a big topic, I would say. So here's the outline of my talk today. So I'll give a brief introduction. So this introduction will be really long because there's a lot of previous work by a lot of major groups that made all those things possible. Which we're building up, just give you an overview of how this would be possible in human brain. And then I will give an introduction of the methods and the application I propose. So those are the methods I developed and their applications here. And those will be fall into 6 major topics. First was the quantification. So not only mapping the human connectome, we need to quantify. We need to see whether connection is good or bad. Then we have a way to study brain disease. Then with the good techniques, we get a very good chartography and apply to clinical application. Also, we need to compare cross subject. Although each individual brain is unique, but we need to come up with a way to compare this patient with that patient. We need to do something called spatial normalization to allow for inter-subject comparison. So that's also a method developed. And with those methods, once we can normalize all the brain together in a common space, then we can take an average and create a template. And it turns out that the template itself also has a lot of application. And finally, with all the quantification normalization, we could have a standard framework to study connectome, which we call the local connectome, and then use it to compare the similarity or difference between them. So just an overview of what we're going to cover today. Any questions before I start it? No. So a brief introduction on mapping human connectome. This slide just shows how challenging it is. At left-hand side is like an MR image of a human brain cutting an axial slice. It's a T1 weighted image, so you get the white regions are the, where the axon connect between brain area. So you get, if you think of the brain as a very complicated circuit, white matter region is like a bundle of axons that are like cables connecting to different regions here. But from MR image, you have no way to tell whether those axons going from here to there or here to a different place because there's a limit in the resolution. The best resolution in MRI is at the scale of millimeter. So it's like a visual you can see by your eye, then that's what you can see under MRI. You cannot see axon connecting from here to there. And so there's a like MR image. You have no way to tell whether the axon is connecting to this region or that region. The only thing that can make this possible is to put tissues under the microscope. So there's a group 3 years ago, they have a project published paper in Science. What they did is really crazy, like fixing a subject's whole brain. So you see the tiny picture is fixing the whole brain and taking consecutive slides one by one from frontal to posterior, and then get all the tissue section out. You see that like a trend of tissue section going out. And next thing what they did is like scan those tissue sections and scan it. And they published a huge database like they call Big Brain. So it's just here just showing like a tiny field of view of one tissue section here. You get under microscopic level, then you started to see, okay, I see the axons going like a super high way connecting between where matter is. Here's the white matter region, here's gray matter region, and you started to see it. So it's just show like if you want to map human connectome, you need something that can tell structure in the microscopic level. That's really challenging. Even though Big Brain is a big breakthrough, but there's two problems. One is it's only 2D. If you cut this way, then you have no way to see the structure of the external connection that going through the plane. That's one limitation. The second thing is you need to open up the plane. You need to kill the subject, but that's not doable for clinical application. So for human connectome project, a big question is there a non-invasive way? We need a non-invasive way to map external connections in human without opening up humans. We open up the brain, detect tissue section. The answer is actually yes, but in an indirect way. So the key here is using water diffusion as a proxy. So even though MR image, usually when we take an MR image, the muscle size is around like the best would be 1 millimeter. And for each muscle we get, we can actually get a distribution of signal that's sensitive to water diffusion. So the idea here is that, okay, MRI, we're not going to be able to see microscopic level of tissue structure. But we can turn on this MRI to sense how water diffuse microscopically within this axon. So here the animation just showed the idea. You see that under microscope, those axons are like water pipe lined together. So they're going in one direction. Those blue dots here is just simulating how water diffuses within or between those axons. So we can see that under those structures, water diffusion is actually following the microscopic structure. So if we have a way to detect how diffusion changes in a different direction, then we may have an idea, okay, at this voxel, we know the fiber is going this direction, not that direction. With this information, maybe we can building up the whole trajectory of external connection. So those things people call it diffusion MRI microscopy, because it has ability to tell us the structure So how does it work? The whole story actually started 50 years ago. So you may really amazed, oh wow, those technology have been through a long time period. But yes, it is, because it's building up layer like a different technology. So 50 years ago, MR physics working on this type of project. So at that time, we don't even have MRI. MRI means we have a mapping of this spatial distribution of water. We don't have MRI, we just have MNR. MNR is just like a scanner, you put a substance in, solutions in, then you get signal out. There's no mapping, no imaging, it's a signal. At that time, there's already a way, so MR physics, what they want to do is, okay, I have a substance, I want to measure something called diffusivity. That's how fast a substance will diffuse within itself. And what they did is, okay, I put the substance in the MR scanner, and then take the signal. So the signal will have an attenuation, the decay, the correlate with diffusivity. If diffusivity is higher, so the substance move around, and then you have the MR signal decrease, then they can calculate it. If the diffusivity is lower, not moving around, then the signal decay is not that great, then they can also map it. So they already developed a way, so a sequence called Stockholm-Turner sequence. Amazingly, this sequence is still used today in clinical scanner. So clinical scanner, when we do like diffusion MRI, they still use this type of Stockholm-Turner sequence. So this sequence allows us to detect the water diffusion and measure something called diffusivity. And after Pathology introduced MRI, then there's a French doctor called Fang-Cheng. He combined diffusion MNR, so those things, with MRI, and introduced diffusion MRI. So the whole idea is, okay, I can quantify diffusivity of a substance. So using MRI, we can quantify diffusivity across the whole brain. So I can feel, okay, this brain region has higher diffusivity or lower diffusivity, and whether they correlated with disease or pathology, and you can provide a lot of information. So it's really a big breakthrough here, and combining those new things together created like a huge application. It's really a milestone in diffusion MRI.
So the picture in the middle, is it telling me the diffusion coefficient of each voxel?
Yes, that's a key point. So each voxel is like the size of like 1 millimeter or 2 millimeters. So you're thinking it's like a cubic. And within this cubic, if you enlarge it, it will be like a system itself. Then the water diffusion inside it, what's the diffusivity inside this voxel could be quantified and then plot here. So higher means higher diffusivity, lower means lower diffusivity. That's the key technology used.
But if the tissue is has some pathology, how will the diffusion change? I mean,
I'll talk about that later. So that's some important aspect I will talk about. Also, one important observation people see from this mapping is that they observe that the diffusivity is orientationally sensitive. That means if you the diffusivity will be different if you measure in x direction, y direction, Z direction. The reason is simple because the axons are lying at different orientation. For example, the Cox callosum with fiber going this way, some fiber may go anteriorly, and depending on your sampling direction, the diffusivity will be different. So ideally, if we can sample a lot of different directions, then we could possibly know where the fiber goes. So then, there's a technical DT I introduced here to answer this kind of question. Say, okay, if I have only limited sampling, can I model it and then calculate what's the orientation of axon? So thinking of within the voxel, the axon could be lying anywhere. The technique here is like taking just 6 sampling and model the diffusion by a Gaussian distribution, a simple 3D Gaussian, and calculate 3D Gaussian distribution and then analyze it, and we know what's the principal direction along the axis, and then that tells us the fiber orientation. The whole thing is made possible in good detail. So this Peter Baser, 20 years ago, proposed the methods, and this method being used in thousands of papers so far. So this just shows you like an example of ETI. So you see for each voxel, you can calculate like a principal direction coded by the color. So to help visualize the thing, the fiber that going left and right will be colored by red, so you see the fibers going in like this direction. Here's the fiber going through the plant, color coded by blue. Some fiber going anteriorly by green. So using this method nicely tells us for each voxel what's the major fiber orientation going. And with those information, we can simulate the trajectory. Say, okay, if I started a trajectory from here, maybe you can connect somewhere to this direction. And maybe we started here, then the axon should be going this direction. So by simulating this kind of trajectory, we can build up a trajectory shown to the right. So the trajectory is showing, okay, what's a possible axonal connection between the human brain. So at that time, this is like really amazing technique. So without opening up the human brain, we can see how your brain connects between brain regions. Also in the, yes?
So I can see how when you're in the corpus callosum and you have all the fibers oriented, let's say right to left, that you'll get a clear vector.
Yes.
But when your voxel contains, let's say, 1,000 fibers, you're really getting the average vector for all those 1,000 fibers. So if you had, let's say, 333 fibers going right to left, 333 fibers going up and down, 333 fibers going perpendicular to that, is your voxel going to give you no diffusivity?
That's actually a really good question. You already get the insight, because like that's a huge limitation of ETI. So people already talk about it. So say for example, if you have two fiber orientation and you model it with just single tensor, it will not be enough. It will tell you something in between. So that's a big limitation. So if I try to point out here, say some fiber over here, maybe there's some crossing here, the fiber orientation can go crazy. So that means two things. One is ETI is not enough. Second thing is this trajectory will not be reliable. I will also cover those two things.
Yeah, maybe on models, if there's a mixture models and
people did that.
people do that.
So anything you can think of, people try to fit with multiple tensor, fit with like a limited degree of freedom tensor, fit with any kind of different model, and try to tell whether there are two fibers, three fibers, or one fiber.
But the colors right now are suggesting, on the left side, colors are suggesting orientation in three-dimensional space.
Yes, yes. So you can see like here is like through plane, green is anterior posterior, red is the right. Yes.
Is this data routinely captured in a garden variety brain MRI, or is this you have to specially capture a raw data set?
There is a sequence called diffusion sensitization sequence, so you have to use that sequence. But that sequence gives you a signal that is close to T2 weighted image. So it's kind of a T2 weighted image, but you have to take multiple set of image just to get this. So imagine that T1, T2 just have one image volume. But if you want to get a whole distribution within the voxel, then conceptually it's a six-dimensional data. And to get this six-dimensional data, you require a long MR scan. So one limitation of the diffusion MRI is the scan time usually is very low. But there are also fast imaging techniques trying to cut that short.
You mentioned the big brain project where
Yes.
like none of the data is useful to cross-check.
The problem is that each individual brain is unique. So for major 1 meter bundle, yes, we can check. But without checking, we know that it should be going there. But the limitation is when they start branching out, and that it has individual difference, and there's no way to check it. Any question? So with this mapping, the second thing people think about is to quantify it. So DTI also offers a way to quantify it. So here is like a 3D Gaussian. So once you fit data with a 3D Gaussian, the Gaussian could be a really long ellipsoid, or just like a ball. They tell, and those Gaussian will tell us the condition of the axon. So say, for example, if we have axons lying horizontally, you can imagine that the 3D Gaussian should fit at the same orientation. If the axon condition is very good, then you can establish a long ellipsoid that tells you this good condition of the axon. If there is some axonal damage, whether it's demyelination, axonal injury, you can imagine the ellipsoid will become isotropic. So there are 3 different indices, or actually 4 indices proposed to do some quantification. One is axial diffusivity. That's a diffusivity along the axonal direction. Radial diffusivity, that's a diffusivity across axon. So people use it to evaluate myelination or demyelination. Mean diffusivity, just take an average of all the diffusivity, just show you the overall velocity of water diffusion. Fractional anisotropy have been used a lot. It's actually a ratio between AD and RD and scale between 0 and 1. So high FA means the axon is really in good condition. That shows a long ellipsoid. Whereas the FA is really small and close to 0 means there could be axonal damage or just no axon at all. So if you search PubMed or Google, it's like those indices, like, you will find thousands of papers using that. We use it a lot in clinical setting, in psychology study, any type of brain imaging study, verify diffusion, probably 90% of them. To just give you a flavor of how those mapping shows in human brain. So here is the FA. So it's not surprising to see that it's high FA in the white matter because those axons are lining really well. But you can see here, there's some crossing between the FA going down. That means it's a poor fit of a crossing fiber. If you have fibers crossing to each other, of course they become even more isotropic. It's not perfectly solid here. Also, you see like the lambda 1 is the axial diffusivity. Lambda 2 and lambda 3 is the 2 radial diffusivity, showing that people said that this intensity correlates with myelination. So a typical diffusion study, what they did is they may recruit subjects, normal subjects, patients, and they acquire diffusion MRI. They calculate diffusivity FA or axial diffusivity AD, radial diffusivity RD, do a comparison, and if it goes up or goes down, then we could have conclusion whether it is axonal injury or demyelination. A lot of studies doing that. Also, with all the local fiber orientation color coded, show as the icon vector, we can also get chartography. But as I mentioned previously, there's a limitation in chartography. First of all, it's simulated. Second, it's not able to tell you whether it's crossing fiber or branching fiber or kissing fiber because there's a poor fit if there are monofiber population there. But we still have something that could provide us a non-invasive way to map. So first question when 10 years ago when I studied diffusion of MRI, the question I had is, are FA and diffusivity good biomarkers? So that's before we really applied it to any kind of setting. We really want to know whether they are good or not. So 10 years ago when I was a medical student, I joined the lab and they're doing diffusion MRI. So the first project I have is, okay, just to see how good it is. So one thing I did is using Fang-Cheng. Because we don't have resource, we cannot open up subject brain. We don't have tissue proof. We cannot just cut tissue and then see, okay, this is some damage. So one thing we can do is using Fang-Cheng. The Fang-Cheng is here is the layout of Fang-Cheng. It's a simple Fang-Cheng. We use capillary tubes that's the size roughly equal to axons, like 10 microns. Line them well in a placeholder made of plastic. And also another capillary tube lying in a different direction, placed in the placeholder. And then over that, then immerse the whole thing in water, just to simulate how brain structure we have complicated crossing and partial volume of water in ventricle, partial volume of gray matter, and do a scan. So in this cross section, you can see that this B region is a partial volume of two fiber population. One is through plane, one is going horizontally. The C is like a partial volume of this horizontal fiber with a placeholder. And A is a partial volume with a free water diffusion, just simulating the water circling around ventricle. And the whole Fang-Cheng put in thermal scanner, scanned it, did analysis of DTI, and just to see whether the index goes well. So ideally, what's expecting is that if I got FA, it should be homogeneously across this fiber, or either across this one. That would be ideal. Or either AD, axial diffusivity, or radial diffusivity should be homogeneous across the fiber. And that would be perfect, and we are ready to use it. But here's the scan, the results I got. You see, here's the FA. There's a significant drop of FA just due to crossing. So that means we don't have to have external injury. A crossing fiber can cause increase of FA. And some partial volume of the placeholder will increase FA. The same thing also happening in axial diffusivity and radial diffusivity. So this tells us one thing. Even though there's no injury, demyelination, or all kind of pathological condition could be revealed by this biomarker, there's still a lot of factors that can bring in a lot of variability. So say, okay, if the difference between normal structure patients is this big, if you have a large variability, you will kill your finding. And this really tells that it could be a serious problem. And if like a technique doesn't work out in Fang-Cheng, of course it will work much worse in human brain, because like human brain, the axon itself, the diameter will be different, the composition is much more complicated than Fang-Cheng. If a technique doesn't work out in Fang-Cheng, we don't have to apply it to human brain. But a lot of studies still using that, just to bring up here that even though a lot of people are using it, it doesn't mean that it's perfect. There's limitation, there's some condition that could make it wrong.
Physically speaking, why does it go down?
So they go down because they fit with single tensor. So once we have two crossing fiber, the fitted tensor would form like a disk. If we form like a disk, then the ratio goes down between the AD and RD and cause the effect to go down.
But if I had a mixture model or something, you think at least in this
It could be better, yeah. So there are people that are trying to, using more complicated model to solve that.
Is this a two-dimensional flattening of the data, or are these voxels like three-dimensional? Like, could you separate out the different phantom tracts?
Yes, so the trick here is I just put a huge slide to cover the whole Fang-Cheng to simulate the limited resolution of MRI. Of course, if we have higher spatial resolution, we can separate that out. But we know that limitation in the MRI is like 1 millimeter, and that happens with, if we are crossing fiber, you will always have partial volume of crossing fiber. It's just to simulate that. So instead of using all the main dimension is using diffusivity, and that has a problem that you can only show the overall diffusivity and it has partial volume problem. So here I try to change the mindset. Instead of using diffusivity, I try to quantify the amount of diffusion. So there's different way to look at the data. Just like, okay, if water diffusion is like a traffic, you can, if you want to quantify traffic, there are two ways. One, you say, okay, what's the average velocity of the car driving on the highway? That's the diffusivity. And also I have another way, and we say, okay, there are hundreds of cars driving on the highway. So it's the amount of water that diffuse along the axon. So it's different mindset. And the good things about this mindset is that if we quantify the amount of diffusion water here, we can easily separate them. So if some water is diffused there, some water is diffused this direction, we can easily separate it out. Compared to diffusivity, you always show the something in between. You only show the overall diffusion velocity. So I'm going to introduce the first method I proposed, developed around 5 years ago. It's a way to quantify things instead of using diffusivity, quantify the amount of water diffusion. So techniques make this possible is based on something called Q sampling imaging. So the contribution I make here is try to derive a mathematical model between MR signal and something we call spin distribution function. So this function is like a 3D orientation function, shows the density of water diffusion. So imagine you have two fibers like going this way, one is vertically, one is like this way. You could nicely showing that there's a peak here, showing there's a fiber going here, and there's another peak here, it's going in a different direction. So compare with DTI. DTI is a model-based. It doesn't matter whether you have one fiber or two fibers, just fit it with one tensor, one Gaussian. But here it's more like a non-parametric way. We can use like a statistical term. It's a non-parametric way to map the empirical distribution of this water diffusion. And with this one, we can have the axonal direction at the peak orientation. We can also use the magnitude as a way to quantify the integrity of axons. So here's just showing the paradigm here. So this one is the SDF, which is the orientation distribution water diffusion. Here are 3 axonal fiber. We can easily take out the isotropic part, which is the free water diffusion. So we have partial volume of free water diffusion, we can take it out. And then we can have quantification, just taking the magnitude to the peak. And it's quantifying the integrity of the axonal fiber here. So how does it work or not work on this phantom? So we have something like a simple ground truth, and we can test it out. And here shows the nice result. So here shows the quantification in the horizontal part. You can see nicely, you can see how nicely the intensity remains the same across the fiber population. So that means the quantity of water is stay the same along this fiber population. Whereas in the vertical fiber, we can also quantify separately and then visualize the quantity of the water that diffuse along this axonal direction. It's much better than the FA. We don't have the partial volume effect. We don't have the high and low different intensity that cause a problem. So with this technique, at that time we are confident, okay, this could be holding the better method to look at human brain. We can get rid of a lot of variability. It should be much better. If you save a tiny little difference, this technique would be a better approach to visualize that. So here's just showing a comparison between FA. FA is based on DTI, which is a ratio between AD and RD. And then this one's a QA. So you can see how nicely, so those are from the same subject, same scan. The data source were the same. Only difference is the way we compute it. So you can see that first of all, the background is very noisy because the diffusivity will tend to be having a lot of noise if the SNR is very low. But here the QA, which is the magnitude on the SPF, nicely showing the clear picture without adding any mask here, so you can see that background is clean. And you can see how nicely they're showing the great white matter junction, whereas the FA, when you're reaching the low SNR errors, getting a lot of noise. And that's due to the fact that there are a lot of fiber crossing when you're reaching the gray matter, whereas the QA can nicely showing the fiber going into the gray matter.
You have to predefine in the spin distribution function how many peaks or how many axonal directions you expect to see.
You don't have, because it's an empirical distribution.
Right.
So it can have different modes.
So you have to go look for the modes. Though.
Yes, I have to go look at modes. It could be 2, 3, or just 1. And thinking of the if you put it into one-dimensional paradigm, DTI just fit a simple Gaussian where there's regardless of data. And the method I use is a model free. So it could be, it's an empirical distribution. You can look at multiple fiber direction and quantify individually. So first thing we can do is use this type of technique to improve fiber tracking. So you see here, if you use FA base, you can get a lot of noisy fiber out here. And the QA base, you can nicely filter out the fiber and then get a clear picture of the local fiber direction to provide for the photography. So if we use this technique to create the photography, so this work with we collaborate with a neurosurgeon at PMC. So what they did is they have like a cadaver brain, and then they scan the cadaver brain and compare with the anatomy. You see how nicely catching those gyral folding, and you see how those fibers entering the greater matter region. And they call this is a high definition fiber tracking, and provide a very good quality of photography, either using circuit planning or any type of application. So just by changing the paradigm, instead of using diffusivity and FA, we use a quantity-based estimation. It could improve a lot. Not just the biomarker, not just the quantification, it also improve fiber tracking. So we did a study which tried to show that this is much better. So what we did is invite some neurosurgeon and then do a blind evaluation. So we have track based on FA, track based on QA. So 2 neurosurgeons evaluate what's the false fiber. So they have like the anatomical information about which fiber is false, which fiber is true, and they quantified it. So the false fiber rate is substantially lower if we use QA compared with other index. It's much higher. You see, like in DTI, FA, like 20% or 30% of fiber is not true. Also, last year, we participate with like conference competition. So there are 20 groups in a row submitting around 90 different methods mapping all the fiber tracks. So our method is like when out in terms of the valid connections, the highest one we can get is over 90%. It's the best among all different other groups. We also release like a open source free package. So any group in the world, you like to use this technique, they can acquire their own diffusion MRI, and then they can run this through the pipeline we provided, and then map the human brain here. And we did get a lot of impact accumulated over the past few years. So starting from 2010, just like 5 papers using the methods. It's like this year we got 70 publications using this tool. Most of the groups we have no collaboration, so they just download the tool, they use it, and then they publish results. Most of them, yes.
Sorry to interrupt.
No.
But how is the other imaging technique that you use to validate it?
It will be very challenging to validate it. So for neurosurgeons, they evaluate based on their knowledge. Based on their experience, when they open up the brain, they have some idea of which region should be connected to each other. They use that as a way to evaluate it. But that's still not a ground truth, but it's much better. Here listing some of the application paper. Some of them use it to quantify connectivity. Some of the groups use it to map brain connectome. Some of the groups use it in brain tumor patients, so it's already applicable to clinical study. So this one study collaborate with the neurosurgeon. They can see, they map how the fiber traffic takes place by tumor. So the good things about this one is like for tumor patient, the doctors could see how important fiber tracks being takes place. So when they have doing the operation, they can avoid it. So if, say for example, some track control your hand movement, you don't want to lose it. So neurosurgeon could approach from different sides. This could be made possible by this accurate mapping of tractography. And all this was made possible by just changing the different paradigm of quantification. So how about deeper comparison between QA and FA? Actually, 5 years ago when I proposed this method, I collaborated with a group. And what I did is like they want to evaluate to see whether it's good or bad. And so their idea is, okay, I have like 5 or 6 normal subjects. And what they did is they have the diffusion MRI scan, and they take out the same region and then calculate the FA and also calculate the QA. So they want to see whether FA is more stable or QA is more stable across the normal population. However, actually at that time, it's really a setback for me. They sent me an email saying, hey Frank, this doesn't work out. You see this QA across subjects is high, much higher variability across normal subject. You see this FA, reliability is much better. Those are normal population taken from the same region. The QA is much having a higher variability. So at that time, I don't know how to explain this one. The one cause I can think of is like because like QA can quantify multiple fiber. It's possible that the QA they took out from each voxel is actually from a different fiber population. And therefore, the comparison could be like apple compared with orange. You taken this fiber for this subject, taken that fiber from that subject. Because they're just looking at the maximum QA value within the voxel. Consequently, it causes higher variability. At that time, I have no solution to that, but this is the fact we're facing. So one idea we think is, okay, maybe we should come up with a way to spatially normalize the brain. If we can say, okay, even though each individual brain is unique, if we put under the same common space, then we could know, okay, the fiber should go this way, and then we can compare the same fiber that going in this direction instead of that direction. So we need something, first of all, spatial normalization for the brain in the common space. Second thing, after this spatial normalization, all the quantifications still remain consistent. That will be very important. So there's the second thing, so the spatial normalization. And it turned out to be very challenging. So just for illustration here, so imagine you have material that's homogeneous. And because we are quantifying density, if we have spatial transformation, it actually changes the density. So the spatial transformation, you have to consider registration between each individual brain, and at the same time, you have to make sure the density remains consistent. So if some area making smaller, density should go higher. If the area is dilated, the density should go down. And luckily, we got a method working that we are able to spatially transformed individual brain to a standard space. So here's just show an example. We put it into the standard space called MNI space. So MNI space is a really popular common space used in the neuroscience field. So here just showing the SDF, the 3D orientation distribution being spatially transformed and scaled into the standard space and align well with the anatomical structure of the template. And these things also guarantee that the estimation is consistent. So that if you have certain region being large, then the density will become lower. The region is getting smaller, the density goes up. And then from this SDF, we can also get the local fiber orientation in the common space. So we have this technique, and the one things we did immediately is averaging all the brain together. So at that time, we are interested in, okay, what's the normality of the brain structure? We had a lot of subjects at that time, like around 100 subjects. What we did is we normalized each brain to a standard space, and we take an average. And what we get is a representative brain structure of a big population. And also, in addition to that, the average itself provides us additional spatial resolution. So we see here, even though the data is acquired by 2.5 millimeter resolution, after average, you can see some structure at 1 millimeter resolution. And with a better data set that acquired at 1.25 millimeter resolution, the spatial resolution much higher compared with the simple T1 1 millimeter resolution. And you see much detail of the track going here, some structure, some tiny nucleus here. So the spatial average not only gives us like a representative brain structure, but also boosting the spatial resolution. That's something that surprised us. So we have this template at that time, and the one thing we did here is use it to study human brain anatomy. So this here we had there was an interesting story about this study. At that time, my collaborator is like a neurosurgeon. He tried to publish a paper using like about 6 cadaver brain, and they would like to study like a structure called NDLF. So there's a pathway connecting from the parietal lobe to the temporal lobe. And the whole paper is really nicely written. It's already in the review process, but there is a reviewers just keep criticizing, give them really a hard time. And the big argument the reviewer gave is that you only have 6 cadavers. How could it be representative to a large population? Say, okay, maybe some spatial structural difference maybe occurring with 10% of the subject population. How could this be representative? So my collaborator talked to me, and we had a discussion at that time. Just came across and I have a discussion, and I said to him, I said, oh, I just recently created a template, averaging from 90 subjects, and we can use it as a framework to do fiber tracking. If we can have a fiber tracking that shows the same structure, and yes, we have it. It's representative now. We have 90 subjects, and we did use that. So I kind of joined this project during the review process, and they use it, and they to track the same pathway. It's nicely showing the same morphology and following the cadaver, totally settle the issue. And then this template is really a really good resource to study brain anatomy. So it continuously published a lot of papers that say, randomly pick out one fiber track, doing cadaver study, doing fiber tracking and template, publish a paper. They did this one and published it continuously, like 6 papers. This year we have 3 additional papers coming up. I said, wow, this is a new combination that hasn't been done elsewhere. It's a perfect match, we could say. Because a cadaver, you cannot do cadaver study on more than 10, just too for the neurosurgeons. But we can do a lot of, we can average a lot of subjects together into a template. The recent template we have is having 880 subjects together, and it's very representative of the normal population. I can use it to study the brain structure, correlate with their anatomy finding. It's really an amazing byproduct by this method development. Also, this spatial normalization or other application by other different groups, those are studied by different groups. They use it to study network analysis. Because once we put all the things into a same framework, they can do a lot of different analysis because they already solve a lot of spatial normalization problem here. This technique can also used to study white matter morphology. So we want to study whether the morphology change or difference, whether it's normal and abnormal in different population. Different groups use that in different applications. So we have quantification way approach, and also we have a nice normalization. Then we can go back to the topic. We don't have to worry about comparing apple and orange. We have a common framework from the template. They tell us, okay, in this fossil, there should be 2 fibers. Then we can have the SDF come in, and we just sample here and sample there. So we know we are comparing this instead of comparing between those two. And it's really expecting whether it gives us a good result. At first, it surprised us. Again, this one is FA. This mapping is the mid-sagittal cut and the corpus callosum. So look at corpus callosum is like a fiber going left and right. It's serving like a hub connecting left and right hemisphere. And you see SDF lying out, and we just take the cut. You see how nice these 3 different subjects. You can see how nicely their sub-FA stay the same across subjects. They're all the same. However, even with those techniques, spatial normalization, quantification, we try to avoid comparing apple and orange. You see this subject has higher here, this subject has a higher intensity here, this subject has higher intensity here. We still run into the same problem. There's high variability across normal subjects. And this FA just stays the same. At that time, we said, wow, is there any problem with the data set? Is there any problem with the computation? How could we still have this high variability that really would kill our finding? So what we did is, okay, try to get test retest. So this is kind of a test retest problem. So we're trying to find out if we have the subject that scanned twice and see whether the test retest reliability is good or not, comparing this example we call local connectome and FA. And then something really surprising showing up. So this is subject 1. This one is scanned after 6 months. The FA is not so different across subject, but what we see is that for subject 1, the same region widened up. So the finding is real. It's not something that's artifact. It's not something that's due to computation error. It's just high quantity of water diffusing over there. Second subject, yes, consistently appearing up. Third subject, yes, consistently appearing up. So we found that this technique, compared with FA, could catch some characteristic that's specific to individual, just like a fingerprint. The fingerprint is unique for each individual. FA does not show this individuality, but the technique we use here show a consistent pattern with each individual brain. So say if you have a scan, then you have your own pattern here. If another subject have a scan, it's a very different pattern. Third subject, then it's totally different pattern. Just like a fingerprint, each individual brain is so unique that they form a high variability. So at first, the variability we see in the QA is not a problem. It's actually an advantage. It's able to show something that's unique to you. And whether this correlates with any biological meaning or any variable would be very interesting. So the things we did is, okay, we map out all those things we call the local connectome across the brain, and we show all the variable up as one-dimensional vector. So imagine like those sampling is across the 3D volume, but we just line up with 1D. You see this is subject 1, you see a pattern. The repeated scan shows the same pattern. Second subject, a unique different pattern. A repeated scan also shows a unique pattern that's the same as the previous scan. And the pattern between subjects is so different that this forms like a fingerprint. So we call this local connectome fingerprint. Local because it's like locally look at a fossil. It's a connectome characteristic, and it's so unique, so we call it a fingerprint. And we were really excited about this result, and we called up to all our collaborators, hey, we will have test result in diffusion line, just send us. So we get a lot of different scans from our collaborator, from UCSB. There's 11 subjects scanned 3 times. CMU, of course, like 24 subjects scanned twice. My previous advisor in Taiwan, they have 60 subjects scanned twice. PITS also had 100 subjects scanned. And they just send me the results. And nice thing we found is like, here the distribution is the inter-subject difference. This one is the difference between scan. So, and this one is the same, the difference between fingerprint of different subjects. And this one is the difference between the repeated scan. You see that those differences are far apart. So we calculate what's the probability of those distributions across each other, and that is the uniqueness of your fingerprint. And the calculation will show that if we acquire repeated scan within 16 days, the uniqueness is very high, the 10 to minus 6. And we have a scan that is longer, 1 year apart, then the uniqueness drop. So this second thing that also surprised us is that the uniqueness drop over time. So at first we saw that, okay, maybe it's scanner issue, maybe scanner degradation. This would be a bad news for us. But if it is real, then we can map neuroplasticity, because it shows how our brain change over time. So we're going to dig into this one. So here's what we show. We have one scan population from the same side that has a different time interval between repeated scan. And here's the similarity between repeated scan. You can see there is a significant dropping trend across time. And also we visualize, so take one subject, and then a year later, you see that this just local region showing up. So this is not due to the overall scan degradation. It's just like maybe we are picking up some neuroplasticity that happens within certain regions here. And it's showing a significant trend dropping. So we're really excited about this technique. Say, if you have a study, want to know whether there's neuroplasticity, we have a way to map it non-invasively. Also, we have twin study. Something also surprised us that we found that for twins, their fingerprints are substantially different, but yet not perfectly matched like a repeated scan. So you see here, even though they're same loci or some small part different. The disjoint twin also the same, even though some same but the difference coming up. Siblings, some similar, but you can see is much more difference coming up in siblings. So we also quantify the difference. Say, okay, here's the distribution between monozygotic twin, this is a DZ twin, this is a sibling, and this one is unrelated. So you see nicely, MZ twin has the smallest difference followed by DZ twin, followed by sibling, and then unrelated subject. So this local connectome fingerprint is not just showing how unique you are, it could also use as a phenotype. So if you have any study that involves genetic, some analysis, you can correlate it, and also we can localize where the genetic penetration is having certain brain region. Also, the second thing we did is we can quantify the similarity between twins. You see the MZ twin is roughly twice more similar than DZ twin. Is statistically significant? Yes.
Is this fingerprinting consistent between scanners?
Yes.
It is. There's no like some factor, scanner factor that's.
There could be scanner factor, especially the signal intensity could be different. But in terms of the framework, so previously I showing there's a framework we use to sample different fiber orientation. We use the same framework there.
So if you scan subject A in one scanner, it's sort of reproducible in another scanner.
We haven't tested that, but we were expecting that there would be very, very different because there will be additional scanner difference also. Yes, that's a very good question. But we haven't tested it out. Maybe, hopefully, we have multi-site study, the same subject scan different site, different scanner. If it shows similar, it would be very cool. So one thing we show here is that this MZ twin is twice more similar than DZ twin, and this match our understanding of genetic contribution. The MZ twin has genetic factor that is twice more similar than the DZ and sibling. And the DZ twin and sibling are there is no statistical significance between their difference. So those things matching our understanding of the genetic contribution in twins and siblings, and we tell us that this fingerprint could be used as a phenotype. So some future works to conclude here. So all those local connectome fingerprints, just like the study we did, we started last year. So there is a lot of things we need to do. So one thing we would like to do is validate it. We see a lot of different intensity across different subjects. So one question we have is, does this correlate with axonal density? Does it correlate with myelination or anything? Because there must be something underneath. So one idea we have is, it seems crazy, but we want to have cadaver brain that is really fresh. Because once you fix tissue, we cannot do diffusion scan. The diffusion pattern totally change during the fixation process. So we want to correlate with some department working on cadaver study, where they recruit a cadaver the same day, taking out the brain and put it into a scanner. Once the scanning done, we do tissue section and this mid-sagittal view, and we map out axonal density. We map out myelination and see which one correlated best. And once we confirm those correlations, we could say, okay, this fingerprint showing us the axonal density. We have a non-invasive way to map axonal density, which is not shown on our other papers here. Of course, we want to correlate with clinical finding if we have clinical application. But tragedy here, we need to have a longitudinal study because the fingerprint is so unique to each individual. It will be a problem for cross-sectional study. We need to have a longitudinal study, scan it the first time, following up, and see which region damage or change over time. That's something we need. Some announcement to my collaborator at different university, they provide me all the data and all the funding source. Thank you for the questions?
Just for a general question, can your technique be applied to other imaging other organs? Or is just, what will be the limitation, for example?
Yes. Another application for diffusion of MRI is cardiac. For cardiac imaging is much more challenging because the cardiac keep beating, but it's still feasible. We haven't really tried that yet, but it is totally doable. But for other organs, we'll be trying, for example, like liver, there are a lot of iron deposition that will kill the MR signal. Kidney is also a challenge.
Like, I was actually thinking specifically about kidney.
Yeah, some people use it in kidney, but we don't have the collaborator working on that, but it's totally doable. We have like a diffusion sequence, and we have a body coil that give us good signal, and of course we can do that.
And one particular application related to the kidney I'm thinking about is, for example, patients who have some forms of glomerulonephritis that are recurrent. One is called renal change disease, so there's a change in the podocyte processes that they retract and then come back. And this would be a beautiful application for patients saving them from getting dialysis.
Oh, yes.
And just having a fingerprint of relapse to remission.
Yes, of course. Especially like kidney, there are a lot of tubules. So ideally, it could be able to reveal the integrity of either microtubule over there, or even
Just the glomeruli, for
Yes.
example, just focusing on
Yes.
one particular area in the kidney.
yes, that would be a good direction.
And well, actually, the glomeruli are affected throughout all of them the same way, so just focusing on one area will window for the kidney.
Yes.
And allow actually using this as a almost like a biomarker of
Yeah, right.
recurrence.
So if you will have like diffusion scan of kidney, you can download a tool and try it yourself. So all the techniques are open. So any group in the world can download it, use it for any kind of. Yes.
Are the voxels, I'm ignorant of MRI, at least parts of it, are the voxels basically how the data is captured by the machine, or is it possible to do things like shift the voxels around or to try to do other things to try to maybe get the resolution higher, multiple scans, or even like a really long scan, since
Yeah.
like that, or
Yeah. So the limitation of the space resolution in MRI is actually defined by the SNR. Because when you acquire MR signal, one step we did is doing the Fourier transform. So we want to have high resolution in the frequency domain. You need to have a bandwidth that's really high in the signal domain. You want to have the bandwidth really high, that means you are able to still get signals when we increase the bandwidth. So there are several ways to increase that. Say, for example, stronger gradient, stronger magnet, try to get that. So say for example, you use 7 Tesla scanner. Ideally, we get a smaller muscle size because we had a higher bandwidth in the signal domain.
But the creation of the voxel units, is that something that's done after the data acquisition, so that the raw data doesn't have the artificial, it's
You're
just a
right.
simple, or it's just a function of the signal, voice signal. Okay.
Yeah, right. Yeah, so there are some group, they even apply like compressed sensing. They will try all different ways, try to boost the spatial resolution. Roughly still at a scale of 1 millimeter, and the best quality will come up from getting the best signal to noise ratio. Yes, of course, yes, we can define the voxel size during the post-processing process.
What's the typical processing time when you have a stack of things to actually?
One subject will be half an hour. Yeah, and we can distribute it and cluster and parallelize it, so it won't be an issue.
What's the typical size of the image?
Without compression, gigabyte.
How long does the scan?
It depending on the number of sampling. Usually, for clinical setting, we now have like 10 minutes scan. It's a decent quality, but if we do get a really high spatial resolution, that could take half an hour. But recently, there are a lot of fast imaging techniques that allow us to cut off the scanning time. Right now, it's like 5 or 10 minutes. We get some quality similar to that.
So these fingerprints are comparison of QA.
Yes.
GFA.
QA. Actually, it's similar to QA, but QA is taken from the peak orientation of the SEM. But those fingerprint, we use the same framework. So the same framework to where this is the direction of the fiber, use that to sample the SDF value. So they are from the same concept, but slightly different. Similar to QA.
So actually, if you want to compare, so this actually ties into the fast imaging techniques. If you want to compare fast imaging techniques, in multiband or whatnot, and let's say you're scanning the same person, and you're using GSI Studio, same Hardy or QBall deterministic model, what would be a good metric to compare the different sequences, like the quality of different sequences?
I see.
Would GFA and QA be?
I think QA will be better.
QA would be better.
Yes. And also we need like a test retest. Similar to that,